This video is about enzymes. So we'll be taking a look at enzymes. First thing to know is that enzymes are catalysts. Basically, a catalyst is a chemical agent that speeds up a reaction without being consumed by the reaction. An enzyme is a catalytic protein. With one exception, some enzymes are made out of ribosome or RNA, like ribozymes. But for all intents and purposes, enzymes are proteins. Hydrolysis of sucrose, as we've looked at in our last topic when we talked about macromolecules, is carried out by the enzyme sucrase. It's an enzyme catalyzed reaction. To break sucrose down into glucose and fructose, we require an enzyme. Every chemical reaction between molecules involves bond breaking and bond forming. The initial energy needed to start a chemical reaction is called the activation energy. Activation energy can be supplied in the form of heat from the surroundings, but not in living cells. The reason for this, if we were to use heat, we would melt everything. So here's an example of bond breaking, bond formation. So if you have hydrogen gas and oxygen gas, you can use it to create water. One way you can do this reaction is to provide heat. An example of this is if you have hydrogen gas and you light a match, it will react with the oxygen gas in the air and form water vapor in the form of steam. Okay, so here is a graphic that shows basically the progress of a reaction over time and what has to happen. So the reactants start out stable. They go through a transition state when enough energy, free energy, is supplied, and they end as products. And the products are also stable. And in this case, the products are more stable than the reactants because they have a lower amount of free energy, or delta G is less than 0. So that's and we'll see that this going over the EA, getting over the activation energy, is what enzymes help us to do. So how do enzymes lower the activation energy? Reactions with more than one reactant, the active site acts as a guide where the substrate comes together in the correct orientation. The active site binds to the substrate, which may bend chemical bonds in the substrate and weaken them. So sometimes the active site actually makes it easier for things to break apart. The active site provides an ideal environment. For example, R groups that are acidic in the active site, which is the acidic environment, allows for the reaction to take place. Now would be a good time to watch an animation. See Google Classroom for the first animation on enzymes. So here's the progress of reaction using enzymes. So the course of reaction with an enzyme, the activation energy, EA, is lower. So it requires less free energy for the reaction to proceed. So we can't have reactions proceed with just adding heat to them in living organisms, because we would denature the proteins and we would denature the very enzymes we require. But we can provide enough heat and then enough energy in other forms for the reaction to occur with an enzyme. Okay, so the environment. Comparing enzyme catalyzed reactions occurring over a temperature range. At low temperatures, the rate of reaction is low as there are few collisions between enzyme and substrate. As the temperature increases, the rate of reaction increases as more collisions occur. At a certain temperature, though, the enzyme begins to denature because it's made of a protein, and above that temperature, the rate of reaction decreases to 0. So as we see we can look at this graphically and see what this reaction looks like. So every enzyme has an optimal temperature. So the optimal temperature for a typical human enzyme is somewhere around body temperature, 37.5 degrees Celsius. Above that, the reaction doesn't proceed as quickly, even though the collisions between the substrate and the enzyme are happening more quickly. The enzyme no longer is functional. The optimal temperature for an enzyme in a thermophilic bacteria, something that's heat tolerant, is going to be much higher. We're going to look at this in the lab. Okay, so increasing temperature increases the rate of an enzyme catalyzed reaction up to a point. Above a certain temperature, the activity begins to decline because the enzyme begins to denature. So here's just a picture of that, what's going on, the denaturation of a protein. So as you can see here, regular protein in a three-dimensional form, denatured protein only has primary form, only has a primary sequence, the sequence of amino acids. So secondary and tertiary structure has been broken apart here. All right, environment in terms of pH. Enzymes are designed for a specific pH. Above and below that pH level, the enzymes will lose its 3D shape and denature. Enzymes have an optimal pH for their environment. So again, the optimal pH for pepsin, a stomach enzyme, is somewhere around a pH of 2, which is what the pH is in your stomach. The optimal pH for trypsin, an intestinal enzyme that's found in your small intestine, is somewhere above, actually neutral. It's above 8, and that because trypsin works in the part of the intestines where bicarbonate has just been released. So it is actually a slightly basic environment. Cofactors. Many enzymes require non-protein helpers for catalysis to occur, called cofactors. They may be bound tightly and permanently or loosely and reversibly like substrate. Cofactors that are non-protein enzyme helpers are things like zinc, iron, or copper. For example, hemoglobin, which you are familiar with as what makes your blood red. Hemoglobin requires iron, and iron is actually what gives it the red color. Coenzymes are cofactors that are organic enzyme helpers. So for example, vitamins. So there's an example of a vitamin there. You don't have to know any specific vitamins. Okay, so substrate concentration and enzyme activity. So as you see, adding substrate increases the activity up to a point. As we increase the concentration for the material, we will increase the rate of reaction. Then at a certain point, no matter how much substrate we continue to add, we're not going to get additional rate of reaction because the enzyme is completely saturated. So you cannot increase the speed of the rate of reaction further than that. So that's our Vmax. Regulation of enzyme pathways. So there's gene regulation. So cell switches on or off the genes that code for specific enzymes. And so specific enzymes, if they're found in the cytoplasm or in the specific area, are going to allow for a finished product. So for example, the starting molecule, you require a first enzyme for the first reaction to make a second molecule. So in here we're looking at starting molecule A transformed into molecule B. You require a second enzyme for molecule C. You require a third enzyme for molecule D. So this pathway only works if you have all of the enzymes available. The induced fit model. So we used to look at models of enzymes and think a different type of modeling. But the most current model is that as the substrate goes into the active site for the enzyme, it actually changes the shape of the enzyme into an enzyme substrate complex. So the enzyme actually bends for the substrate and they work together. Okay. Enzyme inhibition. So it's important that enzymes can be inhibited in some ways, although sometimes this can be a negative. So inhibitors stop the function of an enzyme. Some bind covalent, which is then permanent or irreversible. So if something else binds to the active site, that's a competitive inhibitor. If it binds covalently, that means the enzyme no longer can work. But many competitive inhibitors bind by weak bonds. So a competitive inhibitor blocks the substrate by entering the active site. And that you can see is in this diagram here. And then this diagram here, we're going to talk about non-competitive inhibitors next. But at this point, it would be a good idea to watch an animation of enzyme inhibition for a competitive inhibitor. Non-competitive inhibition. So non-competitive inhibitors do not compete with the substrate directly at the active site. They bind to the enzyme at a different site. So the interaction between the enzyme and inhibitor causes the enzyme to change shape, no longer allowing the substrate to bind at the active site. Toxins and poisons are often irreversible enzyme inhibitors. So example, sarin, which is a deadly gas that was released in Tokyo many years ago, and DDT, which is the insecticide which was released previously to control mosquito populations worldwide. Enzyme inhibition, though, is a part of the normal regulatory pathway. So this would be a good time to watch the non-competitive inhibition enzyme animation. So allosteric regulation. Allosteric regulation is when a protein's function at one site is affected by the binding of a molecule at a separate site. So allosteric regulation occurs when enzymes have two or more subunits, each of which its own active site. The entire complex shifts between two forms, active and inactive. And this really makes no sense unless you watch the animation. So here's the diagram of it. The activator binds at one regulatory site and it stabilizes all active sites into the active form. So there's the activator. stabilizing the active form so that 4 different substrates in this example can bind to this enzyme complex. There's an inhibitor. The inhibitor binds at this same site, allosteric site, and now makes all of them inactive. So allosteric regulation is a form of non-competitive inhibition because it always binds at a separate site. But allosteric regulation specifically refers to multiple sites. Okay, feedback inhibition. So feedback inhibition occurs when an end product in the pathway acts as an inhibitor earlier in the pathway. So this is very useful in living systems. And an example of this here, but really, if you want to get a better understanding, watch the animation. Okay, so here's enzyme 1. And that doesn't matter what the actual enzyme is. Here's the initial substrate. As this product goes through, we form this end product. It turns out, though, that the end product on this is the inhibitor to this initial enzyme. So when this binds in a non-competitive site, it actually turns off the pathway so that no more of the end product can be made. And so here's just another example of negative feedback. So that as enzyme 1 is used to build and go through this process to form product D, it is negative feedback for product A turning into product B. So it actually stops the pathway when lots of this end product is involved. And we'll see this more in depth when we get to cellular respiration. All right, enzyme regulation. This, as well as cellular respiration, the specific location in the cell of enzymes regulates and speeds up reactions. Because the enzymes are located in a complex and actually structurally embedded in the membrane of an organelle, they work much more effectively because one product of one enzyme becomes the substrate of the next enzyme. And so they're all so tightly put together that it makes sense.